Awesome. So, hi everybody. Welcome to the Phi Tau Sigma 2026 research competition. I'd like to first start out by thanking our judges. So we have Deepti Salvi from North Carolina State University, Vishal Maharajantha at Clemson, Zifan Wan at University of Wisconsin-Platteville, Debamitra Day at Antam-Par, Trushan Kumar Shah at Zoetis, and Chetna Shah at University of Connecticut. So I'd like to thank our judges today for being willing to judge, and also I would also point out all the judges were also very instrumental in deciding getting down to the finalists, the six finalists that are here. I'd like to also congratulate our finalists. I would say this is a historically high number of applications that we initially received, and it was very, very hard because there's a lot of really great research to narrow it down to six. So just the fact that all six of you are here is a prize in and of itself. And I'm really, really excited to get to hear about your really awesome research. So this is Phi Tau Sigma, the Honor Society of Food Science and Technology. It honors and elevates the achievements of students and professionals in food science and technology. And so that's what we're here to do today, is to feature some of the really awesome research that our members do. And this competition is sponsored by Phi Tau Sigma. For those who may have the money, definitely feel free to go to our website and donate to support really awesome activities like this. Well, before we go into the actual presentations first, I'll go over a little bit on the format. So each of the finalists are given one slide to present some research that they presented at a conference, and they have 3 minutes to discuss. I'll be timing the different presentations and marking that timing down. So you are supposed to stay within 3 minutes. And then for each presenter, there will be, after the 3 minute presentation, there will be 2 minutes of questions from the judges. And then we will move on to the next speaker. Does anybody have any questions before we go ahead and get started? Hello? Does somebody have a question? No? Okay, cool. All right. So for our first presentation, we'll have Sorour Barekat with the presentation Smart 3D Food Design. Sorghum pea protein gel is a novel structure for enhanced nutrition and digestibility. So Sorour is in the Department of Food Science at University of Arkansas, and presented this work at the American Chemical Society meeting in fall 2025. Feel free to unmute yourself and go ahead. I'll do a little bit of a countdown just so that you know that the 3 minutes has started. Let me just pull up my watch. Okay, so we'll go ahead and get started now.
Hello, everyone. Let me start with a simple question. What if the next sustainable superfood is already grown in our field, but we're feeding it to animals instead of people? My name is Sorour, and today I want to show you how to transform sorghum proteins into high-value food ingredients. Sorghum is rich in non-allergenic and gluten-free proteins, but it has two major limitations. It's low in lysine, an essential amino acid, and has low digestibility. So in this research, we think, what if we combine it with pea proteins, which contains all essential amino acids and is highly digestible? Sounds simple, but there was a challenge. Sorghum protein gel is hydrophobic. It repels water. Pea protein gels is hydrophilic. It attracts water. So when we mix them directly, they don't form a stable structure. To solve this, we use a novel approach, 3D food printing. Using two extruders, we simultaneously printed gels into thin control layers, building a design structure instead of mixing them randomly. In our method, we optimized formulation and printing parameters. Then we evaluated microstructure, rheology, digestibility, amino acid profile, and FDIR, and finally we statistically analyzed our results. Our findings were exciting. We achieved nearly 100% dimensional accuracy. The gels showed shear thinning behavior, which makes them ideal for 3D printing. The microstructure was highly porous, and most importantly, We significantly increase sorghum protein digestibility with balanced essential amino acids. The impact this research can expand sorghum protein applications in alternative plant-based foods, pharmaceutical formulation, and nutraceutical system. Thank you for your attention.
Awesome. Great presentation. So I guess I'll leave the floor open to the judges. Do any of the judges feel free to unmute yourself and ask a question, or if you'd like to type one into chat if that's easier, whichever you prefer. Oh, so Deepti, it seems like you have a question.
Yeah, great presentation, Sorour. Quick question: what was the logic behind combining sorghum and pea protein? Is there like a complementary amino acids, or what was the reason?
Actually, we have two reasons. One of them is sorghum is low in lysine, an essential amino acid. And the next reason was it has low digestibility. In some countries, it's used as a nutritional food, but in the US, it can't use as a nutritional food because it doesn't have all the essential amino acids. So we think if we combine with pea protein, it can complete the amino acid profile. And we also were looking for a structure that we can encapsulate, for example, hydrophobic and hydrophilic compounds. That was the reason we used these two gels
They
together.
are complementary to each other. Is it a complete amino
Yes.
acid source with pea protein and sorghum combined?
Yes.
Okay,
Yes.
thank you.
You're welcome.
Cool. It's a great question. Do we have any other questions from the judges?
Hi, sir. I had a question regarding post-application. Have you looked at how, for instance, if you're looking at plant-based tools, how and where would the pea protein gel find application and what is the economic impact of that?
Yes, in our future research, we use a hydrophobic compound to encapsulate in sorghum protein part, and we use a hydrophilic compound to encapsulate in pea protein parts. And for sure, yes, it has economic benefits because sorghum is not using for human foods. It's mainly used for animals and also even in building materials. So we think, yes, it will.
Awesome. Great question. So it looks like we're up on the 2 minutes of question time. Oh, Chetna, did you have a question? Okay, we have one more quick question from Chetna, and then we'll move on to
Hi, Sorour.
Murphy. Yep, go for it.
Hi, Sorour. Nice presentation. My question is, what will be the next step for your project? You did in vitro digestibility, right? So are you going to do in vivo digestibility, or what will be the next step for your project?
Actually, we are doing our research in engineering lab. We're not going to do in vivo tests, but for sure, if it works, it can be done by other groups in vivo tests. I think so.
Cool. Well, thank you very much for the presentation. That was great work and great questions by the judges. So Murphy, just to give you a heads up, you will be next. Our next speaker, Murphy Carroll, is a presentation called The Development of a Rapid and Minimally Destructive Method for the Quantification of Arsenic in Rice-Based Foods Utilizing X-ray Fluorescence Spectroscopy. He's in our Department of Food Science at University of Massachusetts Amherst, and presented this work at the American Chemical Society Fall Meeting in Washington, D.C. So I'm going to go ahead and get the timer ready, Murphy, just to give you a heads up. And again, if this slide was a little bit weird when I was pasting it in, I think it's okay, but just let me know immediately if it's not, and I can stop sharing my screen.
Okay.
So I will go ahead and get started in 3, 2, 1. Go ahead, take it away.
It may be surprising to hear that arsenic, which is a known human carcinogen, is commonly found as a trace contaminant in rice-based foods. Due to concerns over foodborne arsenic exposure in infants, the FDA recently placed a limit of 100 parts per billion inorganic arsenic in these foods intended for this vulnerable population. This regulatory limit is placed within the FDA's Closer to Zero initiative, which places a large concentration on the development of new and improved testing methods due to the time-consuming, costly, and complex nature of traditional techniques. Therefore, to address these regulations and the limitations of traditional analytical techniques, we aim to develop a method to detect trace arsenic contamination in these foods utilizing an X-ray fluorescence spectrometer, which is a rapid and cost-effective spectroscopic tool enabling elemental analysis in a variety of sample types while requiring little to no sample preparation. To develop the method, we milled and spiked an enriched form of colorants rice with arsenic from a concentration range of about 0 to 500 ppb. Spiked powder was then made into thick pellets, which enabled greatly enhanced detection and greatly enhanced sensitivity in the detection of arsenic when compared to its analysis as a loose powder. Pellets were analyzed by XRF, then to establish a strong relationship between the arsenic concentration in the pellets to their respective XRF spectra. We developed a calibration model utilizing a partial least squares regression tool. Then we aim to further investigate method validity and assess its real-world applications through the analysis of a certified reference material and commercial rice and rice-based foods. Their arsenic concentrations were predicted through the developed method and compared to those results obtained using the reference method in ICP-MS. As we could see, the PLSR model generated a strong calibration with high linearity, yielding an R-squared of 0.99. Further, validation error was less than 9%, confirming the method's predictive capabilities in a rice-based matrix. Most importantly, however, these results translated very well to real-world samples. When we analyzed a variety of rice-based commercial foods, we found that 10 of the 12 arsenic determinations made by the developed method agreed with those obtained using the reference method. This indicates that the method and data modeling tool not only works under controlled conditions, but also is applicable for accurate arsenic determinations across many rice-based food types. These results demonstrate that XRF combined with simple sample preparation and data modeling tools can enable accurate, sensitive, low-cost, and time-efficient screening of trace arsenic in foods. This directly aligns with the FDA's call for new and improved analytical tools, supporting more accessible heavy metal monitoring in these foods and reducing arsenic exposure risk in vulnerable populations. Thank you.
Awesome. Thank you very much, Murphy. I guess, especially because I have conflict of interest, I can't ask any questions for this one. So any judges, feel free to jump in if you have a question or write in chat.
Good job on the presentation, Murphy. Since we are looking into a rapid analytical method, how does it compare to if you're looking at atomic fluorescence spectroscopy or the traditional chromatography techniques?
Atomic fluorescence spectroscopy, in comparison to XRF, in terms of its quantitative abilities, it can also quantify total arsenic, but in terms of its rapidity, still in terms of the sample preparation, those preparation methods for AFS will still be a little bit more complicated than the pelletization method shown here, as well as increased total time with the overall method as well. And then in terms of chromatography, that's used to actually speciate and quantify arsenic in these foods. So in order to use chromatography, you will use that in tandem with another reference method like ICP-MS, which further complicates the entire analytical method.
Thank you.
Awesome. And it looks like Deepti, you have a question as well.
Yeah, nice presentation. I really like that you tested some baby food, it seems like, and see some range, which is good. So can you explain in simple terms, what is your spectrophotometer detecting and how does that reading look like? Is it just a one reading or is it like a multidimensional? And, how did you use that data?
Yeah, so one really good thing about ED or the XRF used in this presentation is that it can simultaneously detect a wide range of elements. Basically, how it works is you bombard your sample with a high energy X-ray. And as those X-rays interact with different elements in your sample, they can kick out inner shell electrons, which are then filled in by outer shell electrons. And that energy loss as those outer shell electrons fill in produce what we call that characteristic fluorescent X-ray emission, which is shown in that bottom left figure. And those characteristic X-ray emissions are characteristic to different elements. So we obtain different emission lines for different elements based on the energy required to eject those electrons.
So is there one peak that you measure value for? Is that right? Or are you looking at the entire graph?
In terms of only for arsenic, we really only investigate one spectral line. So the spectral line we used was about 10.5 thousand electron volts, but you get a range from about 0 to 25,000 electron volts with a lot of different elements.
Okay, thank you.
Yep.
Great questions. And I have one final quick question from Deba Mitra. Can you use this method to detect arsenic from another grain or any other product, or is there there a limited set of products that you can apply this for?
So this method is specifically for rice-based foods. However, we could adapt the method for detection in like wheat-based or other cereal types. However, due to the prevalence of arsenic contamination specifically in rice-based foods, that's why we employed this method for only rice.
Awesome. Well, great presentation. Thank you very much. Sweet. So just giving a heads up and a little bit of a pause here for Menglin to get prepared. So our third finalist, Menglin Han, is in the Department of Plant Sciences at North Dakota State University. She'll be presenting a presentation titled Quantifying Physical Effects in Hyperspectral Imaging, a case study in soybean seeds. This was presented at the 2025 IFT First Annual Event and Expo in Chicago, Illinois. And Menglin, are you okay to go? I'm going to go ahead and cue up the timer.
Thank you.
Sweet. Okay, I'll go ahead and in 3, 2, 1, feel free to take it away.
Hi everyone, today I want to show you how we can see the moisture through the physics. So hyperspectral imaging, or HSI, has become a really powerful tool nowadays because it captures not only the spatial but also the spectral information in a high range of wavelengths. So use hyperspectral imaging, we can do the non-destructively prediction of the chemistry properties like the moisture. But here is the problem. The problem is that when we are doing measuring the soybean moisture using HSI, the lights cannot only, we assume the spectral only mainly reflects the chemistry, but in reality, the light does not only interact with the molecules, it also interacts with the geometry. So let's look at the middle image of this. So when the light hits the soybean surface, the reflection and scattering is strongly influenced by the surface roughness and the packing density. So a smoother or denser arrangement will change the path length of the photons, and this leads to the reflectance shift, not because the moisture changed, but also because of the arrangement changed. The physical arrangement change. So the key insight is this: the light can see the geometry, not only the chemistry. So if we ignore that, our model will lose the noise. To address this problem, we designed a soybean moisture experiment. We created the soybean moisture gradients from dry to 96 hours of hydration, and then scan the soybean seeds using the HSI. At the same time, we use the computer vision to do the packing density, to quantify the packing density, extracting the inner seed distance as a measurable physical descriptor. And this becomes the bridge. So instead of using the hyperspectral data alone, we integrate the density of the soybeans into the machine learning model. So now the model doesn't only see the chemistry, it also understands the physical measurements conditions. So here's the problem. when we apply the physics augmented machine learning model, the accuracy reached the R square of 0.915, and with the 18% decrease of the RMSE, which means the model become more stable under the real measurement variability. This is not just about improving the accuracy. This is about making the hyperspectral sensing more reliable in our real life condition. So to sum up, we can see that the better physics leads to better AI. Thank you.
Awesome. Great presentation. So I'll hand it over to the judges. Do any judges have questions?
So what kind of AI model did you use, and was like, why statistical model cannot describe this? Why do we need AI? Because sometimes people choose AI versus, sometimes statistic can also predict some of the things. So what was your thought behind that?
Yeah, because nowadays we are using the NIR or hyperspectral imaging because the spectral data is high dimension. Yeah, so it's complicated to analyze to use our traditional analyze methods to do the spectral data. So that's why we use the machine learning model, which will be more accurate or speedy. Yeah, and then we compare the several machine learning models, both the tree models and other like support vision support machine SMV. Like we use the different machine learning models to do this. Yeah. Thank you.
Yeah, thank you. So like you saying that spectra, you cannot represent it by one number, to do it in a statistical analysis way. Is that right? Or is spectra different for different moisture content in all the wavelength range, or is there one point that it differs?
Can you please explain the
Yeah, so the
question again?
spectra that you're getting for different moisture content, is the shape very different for each of the moisture content, or shape is similar, but the peak at certain point, at certain wavelength
Yeah.
is?
Okay, thank you for your question. And we can see from the slides, the shape of all the seeds are similar because the the spectra can capture the chemical information. They are at the same range. So the shape is the same, but different moisture will have the different intensities.
Yeah, so in that case, then why not statistical model? Why machine learning? That is my question, because if it's just one peak changing in the height, I know we don't have enough time, but something to think about.
Yeah, thank you. I will think about it.
Cool. Awesome. Well, thank you very much for the presentation. And we'll go ahead and just give a heads up to Carlos that you're up next. So Carlos Porras-Guardado is in the Department of Food Science and Technology at Ohio State University. It will be presenting a presentation titled The Production of Stable and Sustainable Food Colorants from Agro-Industrial Byproducts by Lactic Acid Fermentation. This was presented at the 11th Zamorano Symposium in the US that happened on October 25th, 2025. And Carlos, are you ready are you all set to go and get started? Okay.
Yeah, thank you.
So 3, 2, 1, I'll go ahead and hand it off to you.
I would like everyone to picture a glass of red wine. After months of being in the bottle, that color vibrancy, it's still there. Now, what will happen if you take blueberry juice and you leave it there for some months sitting there? So to your surprise, the color will turn brown. So what's wine's secret? Both beverages have the same pigments called anthocyanins, but in wine, over time or weeks and months, anthocyanins react with other compounds that we refer to as colorless cofactors. And they form a new pigment that's much more stable and also more vibrant called pyranthocyanins. So this is wine's secret. But outside wine, anthocyanins are fragile and they lose their appeal. This is a growing issue because as the US is moving away from petroleum-based dyes, the food industry is looking for a stable plant-based colorants. And anthocyanins seems like the perfect solution, but they just simply don't last enough. And extracting more and more from plant materials isn't sustainable either. So we ask, what if we can give anthocyanins wines stability, but much more faster and using food byproducts? So we turn to blueberry pomace, which is the leftover from the blueberry concentrate production. And as a first step, we did an alkaline extraction that allowed us to extract all anthocyanins and colorless cofactors that we need. As a second step, we did a short lactic acid bacteria fermentation. The lactic acid bacteria cut the colorless cofactor and transform it into a more reactive form. This more reactive form reacts much more faster with the anthocyanin molecule, ultimately forming the pyrananthocyanin as the third step. In other words, we're letting the microbiology do the chemistry for us. This while we manipulate these incubation conditions. And the results are visible. Our extract turned from purple-red in step number 1 to a final red-orange color in step number 3, which is signature of the newly formed pigments. As we can see in the graph, when we apply this fermentation, we have gotten pyrananthocyanin content of up to almost 60% only after 24 hours of formation. As compared to the sample that didn't receive fermentation, we didn't see any pyrananthocyanin. This means we can turn food waste into vibrant and stable food colorants in record time. By borrowing a trick from nature, we are providing the food industry with the next generation of sustainable and stable plant-based food colorants. Thank you.
Awesome. Great talk. Is there any questions? I guess I can start with the question. So once you get this pyrananthocyanin, how stable is it? Like in terms of like, have you done like shelf life studies? Is there a risk of it potentially fading or reverting back to a different form?
So we have done in our lab a lot of testing to the stability of these compounds towards heat, also towards some other ingredients that like as ascorbic acid that fade the anthocyanins but doesn't fade the pyrananthocyanins. Also heat will degrade the anthocyanins but does not degrade the pyrananthocyanins. So they are much more stable than the anthocyanin precursor.
Cool. And I guess, oh, yep, go for it.
Sorry. Great narration. Yeah, colors is a huge thing in the food industry currently, and I'm currently working on various color projects for my company, and it's very challenging, even with anthocyanins. So my question, which is a common problem with most of the natural colors, would be basically you're trying to concentrate something. So the higher the concentration in most of the natural colors, it leads to off flavors, which is very common in anything which is extracted from similar to other anthocyanin sources, could be beet, radish. Have you looked into the off flavors in this, and how are you trying to mitigate that?
So, well, that's a really good question. Yeah, like for example, we look like a red radish extract. Like it has like a strong off flavor. But the thing is like also the extracts come in like a very concentrated, in a really concentrated form. So at the end, the actual amount you're putting in a food product, it's kind of minimal that it won't affect the color of the flavor of the final product. But that's just what I'm kind of like thinking, and also what some other companies have also said to us, but I haven't tested in particularly that aspect in my research.
Have you looked at the intensity of this color to red 40, which is a common thing used to get the pink and red pigments in the food industry?
That's a really good question. So far I haven't, but that's kind of like on my list right now since red 4 is also kind of like a red-orange hue. So
Yeah.
just kind of like a side comparison. Yeah, that's
Yeah,
it.
that's just something to think about because if you're using red 40 at 0.05%, some of these natural colors you might have to use at 1 to 2%, which is a really high concentration,
Yeah,
which can contribute a lot to the flavor of the product.
that's
Thank you.
another challenge. Yeah, thank you.
Yeah, I have one question. I know you said the control was no bacteria, but was it acidified, because it seems like acidity also helps. So
Yeah, so the control received all those changes in the acidity and the same treatment, just not the fermentation itself. Yeah.
yeah, thank you. That explains.
Cool. Great question. And thank you very much. Great presentation, Carlos.
Thank you.
So we're at time, so we're going to move on. So Harmeen, just giving you a heads up, you'll be the next presentation. So Harmeen Prashar is in the Department of Food Science and Technology at Ohio State University, and we'll be giving a presentation called Piranoanthocyanins Surpass Efficiency of Anthocyanins and Reduction of Inflammation Under Hypoxic Conditions in Vitro. So continuing on the piranoanthocyanin note. And Harmeen presented this at the 2025 IFT First Annual Event in Chicago. So Harmeen, are you ready to go ahead?
Yes.
Okay, so I'm going to go ahead and hand it off to you, and I'm going to go and start the timer now.
Hi, I'm Harmeen Prashar. When we think about food color, we often picture bright, eye-catching colors, colors that make food look fun and appealing. But long before artificial colors existed, color in food came from plants. These natural colors are not just decoration, they are bioactive compounds, meaning they interact with our bodies. One of the key processes they influence is inflammation. Inflammation is a quiet but powerful force behind diseases like cancer, cardiovascular, and neurodegenerative. For me, this isn't just academic. I lost a loved one to an inflammation-related illness, and that experience continues to shape the questions I ask as a researcher. So I ask a simple question. Can natural food colorants help reduce inflammation? The blue-purple colors in blueberries and blackcurrants come from natural pigments called anthocyanins. They are known to be healthy, but they are also fragile and unstable in real food. There is a more stable version called piranoanthocyanins. They form naturally in aged red wine and keep their color much longer. What we did not know was, could they also work better in the body? To find out, we tested pigments from blueberries and blackcurrants in cells exposed to conditions that trigger inflammation in cancer-associated fibroblasts and brain tumor. Here's what we found. Blueberry piranoanthocyanins reduced inflammation the most. Blackcurrant pigments, even the stable ones, did not show the same effect. This tells us something important. Not all colorful foods and pigments are the same. I like to think of inflammation like a building on fire. Anthocyanins are like firefighter with a bucket. They help, but only a little. Blueberry piranoanthocyanins are like a fire truck. They have the power to calm the fire much more effectively. Natural food colors from blueberries may do more than make food look appealing. They may help protect our health. So next time you see something colorful on your plate, ask yourself, can it do more than look good? Because when it comes from nature, it just might do more than look good. Thank you.
Awesome. Great talk. I guess, do any of the judges have questions? Okay, I guess I can open a question. In terms of looking at the berries, are there other pyranthocyanin sort of potential sources that you think that you might want to look into, or what was the rationale specifically for just blackberries and blueberries?
Yes, thank you for your question. So the anthocyanin structure from blueberries and blackcurrant is the key. In blueberries, the B ring of anthocyanins, they contain methoxy group and hydroxyl groups. So literature showed us that hydroxyl groups are the key for antioxidant activity. So the rationale was more of the antioxidant groups on the rings, more substitutions of hydroxyl groups could contribute to more anti-inflammatory activity. So I choose blackcurrant against blueberries, which has more hydroxyl substitutions on the rings. But surprisingly, even the more hydroxyl substitutions did not show the same effect like blueberries. Then what makes blueberries different? What gives them edge? That was methoxy group on their B ring. So for the chemical compounds to interact with the cellular targets, they need to have a little bit of lipophilicity in their structure. Methoxy group provides that lipophilic structure, which helps pyrantoanthocyanins to enter the cellular targets. That's the key. And we are still looking into different substitutions on the B rings and E rings and learning more about what makes the key.
Well, yeah, it's a great, and I guess it's like a follow-up though. I was just kind of curious, and I'm not in nutrition, but so like you take these, do they potentially get chemically modified in the entire digestive process before they get absorbed, or is it pretty stable?
So this is a very foundational project. We are growing from here. That are the next steps to look for bioavailability. I am doing in vitro studies to test more about bioavailability. Our next steps are to carry out in vivo studies. And that's the question in my mind, and I'm going to look forward to answer that in my research.
Awesome. I guess, do any of the other judges have time for, for a quick question or? Okay, cool. Well, thank you very much. It was a great presentation. We're coming up on 2 minutes right about now. Thank you very much.
Thank
All
you.
right. Awesome. And so for our final presentation, we'll have Pratiksha Shrestha in the School of Nutrition and Food Sciences at Louisiana State University, who will be giving a talk titled Antimicrobial Nanocomposite Films Incorporating Niacin and Oregano Essential Oil for the Safety of Low Moisture Food, In Vitro and In Vivo Assessment. Pratiksha presented this at the International Association for Food Protection's annual meeting in Cleveland, Ohio in 2025. Pratiksha, are you ready to go?
I'm ready.
Cool. All right. Well, I'll hand it off to you and start the timer now.
Foodborne illness is a serious concern worldwide, and there is increasing concern for ready-to-eat, packaged, low-moisture food due to numerous outbreaks and recalls associated with salmonella and listeria contamination. Although several processing methods can be applied for safety of these food, post-process contamination can still occur during handling, packaging, and storage. To address this problem, my research focused on developing an antibacterial nanocomposite film that can slowly release antimicrobials over time. First, the antimicrobial film was produced using solution casting method incorporating natural antimicrobials, essential oil, oregano, essential oil, and niacin. Next, to evaluate its in vitro antimicrobial activity, the film was punched into 9 mm disc and placed on agar plates inoculated with outbreak strains of salmonella and listeria, and cleared zones around each disc was measured as its antimicrobial activity. The maximum zone of inhibition was 19.5 mm for salmonella and 25.5 mm for listeria. Principal component analysis also indicated the film's greater effectiveness against listeria. To evaluate the film under real packaging conditions, fresh pecan kernels were separately inoculated with outbreak strains of listeria and salmonella. The pecans were then packaged in SDPE pouches containing the antimicrobial films and stored under controlled conditions. The sample were then collected at different time points from day 0 to day 120 to monitor the antimicrobial effects and survival over time. The results showed that the film demonstrated a gradual reduction in bacterial counts over storage time for both organisms, achieving significantly greater reductions than control films without antimicrobials. And to further confirm antimicrobial integrations into our film, various physical and chemical tests were performed. Microscopic images revealing smoother fibers in a antimicrobial film supported the integration of the antimicrobials into our nanocellulose matrix. For the FTIR spectroscopy of the film confirmed the antimicrobial incorporations as we detected the characteristic fingerprint regions corresponding to both antimicrobial niacin and oregano oil. And in conclusion, yes, natural polymers-based packaging material incorporating natural antimicrobials can be made to effectively inhibit foodborne pathogens such as Listeria and Salmonella. And this approach shows strong potential as a sustainable post-process interventions for safety of ready-to-eat, packaged, low-moisture food and can prevent foodborne illnesses. Thank you so much for your attention. If you have any questions, I'll be happy to answer.
Awesome. Great presentation. I guess, do any of the judges have questions for Pratiksha? Okay. Well, I have a question. Oh, go for it.
I had a question. So you looked at the essential oil, correct?
Yes.
Did you compare multiple essential oils? Because again, depending on the source and the cultivar, the efficacy could be different. And also the concentration of the bioactive constituents play an important role. I think oregano, you might find carvacrol, thymoquinone, and few other bioactive components. And this could be vary based on the source of the oil or the type of, is it cold pressed oil or a different extraction method is employed. Did you compare multiple oil sources and compare with the standard?
Thank you for the questions. I only took the essential oregano essential oil that contains 70% of the carvacrol. That was the major antibacterial compound in it. I chose that oregano essential oil because my target was to have the antibacterial film that could have the broad antimicrobial spectrum. And the literature review showed that the oregano essential oil was effective in inhibiting gram-negative bacteria such as Salmonella. So I chose for the oregano essential oil.
Follow-up question regarding the testing. I suppose you also tested MIC for this, or did you just base it on the zone of inhibition? If so, based on the results, do you think it would have the same effectiveness against different strains of Salmonella or Listeria which you tested?
I did not do the minimum inhibition concern like MIC. I did not do. My result was based on the zone of inhibition, and on the basis of my experiment, for different outbreaks of Salmonella and for different outbreaks of Listeria, so different antimicrobial effect against those antimicrobials.
Thank you.
But the inhibition for the Salmonella and inhibition for the Listeria were showing like similar trends. And when we compared the inhibition for the Listeria and Salmonella, Listeria showed greater inhibition, showing that the film was more effective for the Listeria.
Thanks.
Okay, and I see Chetna has raised her hand, and I see also Deba Mitra has some questions. I know we're limited to 2 minutes, but if we could maybe just really quickly answer them, I guess. Chetna, did you want to go ahead and ask a real quick question?
Sure, yeah. It's a great presentation, Pratiksha. My question is regarding aroma of specifically oregano oil, as it has a strong flavor. Do you think it will affect during storage the aroma of the essential oil?
Thank you for the question, Chetana. So we also are concerned, that is the most concerning thing in my study, because since I have tested in the pecans and it's been more than 120 days of storage, and from the sensory evaluations, we haven't experienced the rancid odor. but we have planned for those proper sensory tests for in our upcoming days to see like how the consumer would react to the aroma in the pecans, and would they be happy to have those flavor in the nuts, or like how willing they will be have those flavors in the products.
And for our final sort of quick two-part question, so how did you prepare these films, and what sort of future, you just mentioned that you'll be doing sensory testing. Do you have any other future work that you plan for this project?
Yeah, also like I haven't showed many of the works that I have done because of the time limitations. I'm also side by side planning to study the antimicrobial release kinetics from the film, because like so far I know there is little inhibitions from these films to successfully inhibit the post-process contaminations, but I still don't have the answer like how the antimicrobials are releasing over time. So I'm planning that study, and also like I will be doing the SEM, further SEM study to see like how the bacteria is affected with the antimicrobials. The literature says that nicene have the pore forming effect on the bacterial cell. So, but I will also be willing to see like what would be the effect on the bacteria cells itself with the nicene and oregano oil.
Awesome. Well, thank you very much. Great presentation. So that concludes the presentation portion of our competition. I'd like to again thank all 6 finalists as well as the judges. So what we're going to do now is the judges are going to go into a breakout room to deliberate for the winner. And in about 10 to 15 minutes, we'll reconvene and announce the winners of the competition. I'm going to stop the recording now. So for those of you watching this after the fact, please join our website at phi-tau-sigma.org if you're interested in learning more about the Honor Society of Food Science and Technology, as well as feel free to look me up on the UMass webpage and email me if you have any questions. So I'm going to go ahead and stop the recording.